here we are in a more specific session, and therefore we are really on the neck of the hourglass. So the methods is, this is a sentence, for instance, 100 male and 100 female undergraduates were shown one of two movies. So this is a very broad methodology, but it's just to let you know that the methods you need indeed to describe what did you do in order to get those results. So which is the technology you applied, which is the methodology you applied, the protocol procedure you applied, and the statistical test that you applied in order to draw the conclusion that the results are significant. And in terms of results, you need indeed to describe what you get. Table 1 shows that the men in the father watching condition cried significantly more. So what is the outcome of your investigation? What did you get with those methods that you applied to your study? So this is a very specific part that is unique to your article and unique indeed to the novelty that you are highlighting in your manuscript. The method, and this is very important, should allow other colleagues from all around the world to replicate your study. And the answer to the question, how? How did you do that? This session explains indeed how you have obtained those results. So, what has been done? What did you look for? How was it done? All research is expected to be reproducible, which is why this section is particularly important. You need to include all the details that allows others to repeat the experiment if they wish to do so. Give the sources of material, the brand and the make and model of the equipment, the quantities that you use from the stock to work in solutions, the duration of your experiments, the reasons that you apply the specific reagents, etc. Describe all the treatments that were done and how you arrange for appropriate controls. So valid comparison that can be made afterwards between the comparison sets. And this is the easiest session to write. It's a good idea to start your writing with this session. And this is an advice that we are giving to all the students because you are bound also to write some, your experiments and your protocol procedures on your lab book. So while you write on your lab book, try to write in the best possible way. So then it's easy to simply take those texts and just put it in a way that is accepted then in a journal article. So this is why is the easiest section to write, because it's something that you already written, okay, either in your lab book or in your computer. So what to include in the method section? Enough detail so the reader can understand how experiments were done. Use figure for clarity. And here I'm making an example for you to understand that is not the same figure that then you will insert in the results. But for instance, if you are using a flow cytometry to address a specific question, you might want to highlight the strategy in terms of gating that you are using to then get those results in the methods. So the strategy is something, the results is another thing. So in these terms that sometimes it's easier, right? So provide the details version of procedure in supplemental data if you don't have enough space for writing everything in the core of the manuscript. And then also state the funding sources and adherence to institutional review boards guidelines if that is needed because of specific experiments that you are conducting. Sometimes it's also useful to introduce tables in this session if you need to, for instance, describe primers is good to put it on the on a table, or if you wanted to provide a list of antibodies as well, is more useful sometimes to be seen in a table. Because those session are statement about what you already did to answer your specific scientific questions, so then the meta session is always written in the past tense. So use simple past tense. The passive voice is quite common, like the essay was performed, but the active voice is also acceptable. We performed, we evaluated, we developed. And the present tense is used only when describing how data are presented in the paper, because the information is still true, meaning that the data are summarized as mean plus minus standard deviation and so on. So that is the only way to introduce indeed the present tense. And in this, so we are highlighting these because the present is also for something that is indicating that the data still stand, and this is why that if you say the data are the experiment was done in triplicate and the data are presented as mean plus minus standard deviation. So these are essential checks for the meta section, meaning to be specific and detailed as possible, facilitating the reproducibility of the experiments, give information on the study design, the sample collection, the selection criteria, the ethics, trial registration if you are conducting one, and the consent details wherever is applicable, the procedure that needs to be very well described, and use diagram, flow chart to facilitate the description if necessary for design, and include the data analysis session to explain the choice of analysis and statistics to answer those research questions. And here I'm really emphasizing the importance of statistics because reviewers are really checking that you apply the right test to indeed elaborate those conclusions.